The IC3BB complex. This is the same C3b as you saw there at the end, which was cleaved by the soluble C3 convertase, and then one part of the C3b was fixed to the surface of the pathogen. And then I told you the rest of the events would be the same. So these are the same actually. Factor B is coming here, attaching to C3b, and C3b, when it attaches to C3b, it undergoes a conformational change. This is detected by factor D. This is an invitation letter for factor D to come here and bind here to this factor. And it would, in the same way, cleave this into two parts, the smaller and the bigger part. The bigger becomes attached to it. And this is a C3 convertase, a potent C3 convertase, because this is not IC3b. This is C3b, okay, and BB. So C3b, BB is a potent C3 convertase which can now cleave more C3 molecules like this and turn it into smaller and bigger parts, and the bigger parts, all of them would be fixed to the surface of the pathogen. So lots of C3b molecules would be generated as a result of this action. Now these molecules which are fixed here on the surface of the pathogen can again engage a number of factor B molecules which would come here and make these C3b molecules into a C3 convertases, okay? And these C3 convertases would continue to do their job to cleave more complement 3 molecules so that more and more fixation of these molecules would take place on the surface of the pathogens that are there in the surrounding, okay? So this is a very efficient process of the innate immune system to mark or coat pathogens in this way. And they are coating them for what? For identification and for destruction, okay? So that these pathogens are recognized and destroyed. Now, this process is not, you can say, a random process, something or some mechanism that goes on inside your body should not continue for, forever. It should come to an end at a certain point. Otherwise, if this process goes unchecked and it is not stopped somewhere, at some point, then all of the reservoir of C3 molecules, for instance, or other complements made inside your body would be replenished. They would just, these complements would be used up in this process, leaving nothing for other pathogens if they infect you. So all these different processes are very well regulated, and for regulation of the C3b, there are some mechanisms in place, For regulation of these, the C3b activation or inactivation, the plasma proteins, both plasma protein and cell membrane proteins are used inside our bodies. 1, 2, and 3, properdine, factor H, and factor I, these are plasma proteins. And DAF and MCP, these are cell membrane proteins. And all these proteins are involved in regulating the production or activation of C3b. Now, what is the properdine doing? Properdine, this plasma protein, as you see here, when this is a C3 convertase enzyme, C3b is bound to the yellow factor BB, okay? So this is a potent C3 convertase. This can convert C3 into A and B. But this assembly, C3b, BB, at the top, it also is prone to be attacked by some proteolytic enzymes. And other proteolytic enzymes can just come here and cleave this structure so that this convertase may no longer be functional. It may not be, functional in order to cleave more C3 molecules and make more convertases. So this property, it provides stability to this structure, the C3b BB convertase. When this pink, peptide or property, it comes here and binds to the C3b and BB, it becomes more stable and more active. In this stable structure, always the stable structures would give you more yield. Okay, it is stable. It has. It's not scared to be destroyed by anybody now, and now it is more efficient to do its job. So this stable structure is going to cleave lots of C3 molecules in order to make C3a and B, and the B parts in the same way as I told you in the previous slide would be fixed to the surface of the pathogen. So if more and more C3b are generated by this structure, which has been stabilized by property, it is a kind of activation of C3b molecules. But if this process continues, as I told you, all the resources that you have, all these complement factors which are there, can be depleted in the body, So there should be an end point to this process when it is not needed or not necessary to fix more complements on the surface of pathogens, or enough is enough. Whatever was needed already has been accomplished. So now the process has to stop. And for that purpose, there are other proteins or peptides inside our bodies that are doing the same thing. For instance, this one, an activation of C3b by factor H and factor I to give fragments IC3b. Okay, this is C3b, and this is the same C3b as has been fixed here on the surface of the pathogen. Okay, and this C3b has choices. It has choices to bond either with factor B in order to make a functional C3 convertase, or it has a choice to bind with other factors like factor H. So factor H, it comes here, C3b prefers to bind with factor H, and factor H, when it is bound with factor H, the C3b undergoes conformational change as indicated by the groove in that C3b molecules. And this conformational change activates this factor, factor I. Factor I recognizes this conformational change, would come here and bind to it, and cleave this C3b into pieces. Okay, it would just take a part of the C3b with it, and also dissociating this kind of assembly. Factor I doesn't like this assembly at all. Factor I thinks that this is some kind of a unnatural combination or unnatural union of people, okay? And this assembly should be dissociated. And for that purpose, it gets a perfect chance here in the form of this conformational change, where the factor I is coming, attaching at or attacking at the right spot, and not only cleaving this particular C3b into fragments, IC3b, a little bigger fragment and a smaller fragment, but also dissociating other factors to do the same thing again. Okay, so IC3b is also fixed to the surface of the pathogen, but this IC3b is not competent enough to recruit BB, okay, or to recruit factor B. So this is not qualified, or this IC3b is not qualified to marry BB, okay, or the factor B. It can remain in solitary confinement here on the surface of the pathogen. Although it has some function that I would tell you, but it cannot perform the same function as intact C3b can do. In fact, C3b can make C3b convertase, but this molecule cannot do the same job. So we call it inactivation. No more fixation of C3b, no more coding. And in this way, this kind of C3b activation, it is regulated. Now some people, they have deficiency of factor I, this particular factor which is doing this job. This deficiency of factor I would result in if this already I have told you about this information, that if the C3 convertase activity goes unchecked, then there would be depletion of the C3 in the blood, lymph, and extracellular fluid, and that actually would lead to less fixation on the surface of the pathogen. And if there is less fixation on the surface of the pathogen, it would lead to less efficient phagocytosis. If there are no more C3b, or there are less number of C3b molecules attached to the surface of the pathogen, the process of phagocytosis would be less efficient. If there are no C3b attached to the pathogens, even then phagocytosis takes place in case of many different kinds of bacterial species. But there is a very insufficient, inefficient process that you can say. So this lack of factor I would cause this genetic predisposition to certain infection, for instance, especially infections with encapsulated bacteria, because encapsulated bacteria have this quality that these bacteria resist phagocytosis. Okay, and how the body is dealing with that situation, the body is fixing more and more of C3b on these encapsulated bacteria in order to just get rid of these bacteria, in order to mark them or cover them, or to be taken up by phagocytes. But which one?
Already cleaved by C3b complex. The one with yellow BB.
Yes.
The H cannot bind to that complex, or it's just gonna bind to C3b.
It only binds to C3b, or it can bind to some sialic acids that I would show you in the next slide, and it cannot bind to BB directly. Okay.
Directly to that complex.
Yeah, it cannot bind to that complex because these molecules or peptides bind to very specific places. You would have heard about many peptides that are binding to specific sequences. For instance, different transcription factors in your body, which would bind very specifically to certain sequences, but not all other sequences. There are many endonucleases that you know of. These endonucleases either would recognize 4 base pair or 6 base pair sequences. We call them 4 base pair cutters, or 6 base pair cutters, or 8 base pair cutters. And that is a specific sequence of nucleotides that these enzymes would recognize and cut the DNA right from there. Okay, so the peptides are very specific in doing their job. They're not going to bind to any other structure. Yes.
C3b है ये नीचे वाला ना, इससे H bind करता है. तो इसके पास में चॉइस है कि ये BB के साथ bind कर सकते हैं, H के साथ तो फिर कुछ नहीं, H के साथ क्यों bind करें?
क्यों बाइंड करता है? This is, ये आप C3b से पूछें कि why. This is a regulatory mechanism. As I told you that this, marriage is not a happy marriage, and it results in this thing, okay? And he or she actually had committed a blunder. And this is the price that he or she paid here, okay? So this is a regulatory process that is happening there. although there is a choice, but variability of factor B, for instance, at certain point of time during this process when the infection is proceeding, it depends on the availability of these factors, that at what point of time which factor would be available in that particular microenvironment. Okay, so at the end of infections, when the threat almost has been neutralized, these factor H, the number of these factors or the quantity of these factors may increase in that site where infection is going on. And that's why there are chances for factor H to bind with, C3b, although factor B can also bind with C3b, but factor H can also do it, okay? So somehow this factor is mistaken here. And it happens most often that people are mistaken when they are going to marry someone. Have you heard about it? Okay.